So today we're going to get started into chapter 19, but before we do, we're going to talk about the last topic that is listed in chapter 18, which is the Stork-Enamine synthesis. After Gilbert Stork, a fairly famous organic chemist from many years ago, who discovered this reaction. Can anybody tell me what the reaction is? It utilizes an enamine, but it is not the synthesis of an enamine in and of itself. How do we get an enamine? That's right. So if we take a ketone, and I'm just going to put an R here, okay? And we react that with a secondary amine, and in the enamine synthesis that Stork used, they start off with piperidine, and it turns out that that's a secondary amine, reacts with an aldehyde or a ketone, and we get an enamine. Not an imine, but an enamine. What does that look like that we've been talking about in chapter 18? Pardon? Almost. An enolate. It kind of looks like an enolate. And why does it kind of look like an enolate? How does that look like? What's similar? That's right. So we've got a heteroatom attached to an alkene, just like we have here, right? We have a lone pair of electrons on this nitrogen, just like we have lone pairs of electrons on the oxygen. So if we look at this enamine, how would you expect it to react? What will it react with? What have our enolates been reacting with? Huh? They can react with carbonyls. We're going to talk about that today, though. What have we been doing with enolates? Matthew? Okay, like what stuff? Okay, how do we make a carbon-carbon bond with an enolate? What do we use as the reagent? We have an enolate. What else do we add? What is the enolate functioning as, an electrophile or a nucleophile? A nucleophile, right? So it's got to react with some type of electrophile, right? And if you're going to make a carbon-carbon bond, what's a good electrophile that contains carbon? Other than a carbonyl, which we'll talk about today. Alkyl halides. That's exactly right. So we can take an enolate, right? And we can react those with alkyl halides. And we can get what we call alkylation reactions to occur, making new carbon-carbon bonds between the enolate, this carbon of the enolate, right, and the carbon of the alkyl halide. The same thing happens with enamines. They will react with alkyl halides, very similar to how an enolate reacts. Now what do you think happens? Does that look stable? Why not? Nitrogen has a formal charge. So what do you think will happen when we work this molecule up by dumping it into water and maybe with a little acid around? What do you think is going to happen? That looks like an imine, right? Except it's an iminium because it's positively charged. So what's going to happen when I place that in water? And what do I replace it with? And oxygen. That's right. So it is a mild way in which we can actually do enolate chemistry without actually having to generate enolates using strong bases. Okay? So if we were to start off with the ketone and not go through the enamine synthesis, we'd have to generate what enolate, the kinetic or the thermodynamic enolate, if I'm going this pathway. The thermodynamic enolate, and so what base would I need to use? I could use LDA, but sodium hydride would be better. LDA, I would have to let it warm up and do a bunch of stuff, right? But sodium hydride will generate the thermodynamic enolate, right? So I got to use a strong base to promote that. Is this secondary amine a strong base? No, it's not. And so this will react with the ketone forming the enamine, and then the enamine will react like an enolate to ultimately give me my product. It's a much milder method of doing enolate chemistry, and it is a reaction you need to know. Okay, the Stork-enamine chemistry. So this is not a strong enough base to generate the enolate. That's the important point. And this is what a lot of students mess up. They go, well, that's a base. It'll abstract that hydrogen and generate the enolate. That's not a strong enough base for that to happen. But it generates an enamine which will function like an enolate. Does that make sense? I see a lot of, it's Monday morning, I know. Huh? Exactly what? It's Monday. Chapter 19, we're really still talking about enolate chemistry. Okay, you're going to learn some new names to reactions, but the mechanisms are essentially the same. On exam 3, what reactions do you think are going to be a lot on exam 3? Enolates. Okay? And the chemistry doesn't change that much, but the names do. So keeping track of the names of these reactions is going to be a challenge for you. Okay? So think about how you can organize that best for your learning style. Today we're going to talk about the Claisen reaction, the Diekmann condensation, the malonic ester synthesis, and the aldol reaction. Hopefully we'll get to the aldol reaction today. Okay? So let's do a little thought experiment. Let's suppose Jamie has a bucket full of ethyl acetate. Okay? What's the pKa of these alpha hydrogens for an ester? Approximately. About 25. What's the pKa of sodium ethoxide as a base? Is it higher than 25 or is it lower than 25? Higher? Sodium ethoxide is more basic than that? It's lower, and a little bit or quite a bit? The reaction does go, but the rationale's not right. What's the pK of an alcohol? 16 to 18, right? So we're at about 16 to 18 for that base, and we're at about 25 for the ester. That's a big difference, right? So is the equilibrium in generating the enolate from that reaction going to lie to the left or to the right? So am I going to generate a bunch of enolate or very little enolate? You're supposed to tell me. I've got a base that has a pK of around 16 or 17, and I've got an acid that has a pKa of 25. Is that reaction going to go to completion or not? And you would be correct. It will not go to completion. For the reaction to go to completion, remember your base has to have a higher pKa than the acid, right? So here we have a base that has a lower pKa than the acid, right? So do we generate a little bit of enolate or a lot of enolate? Just a little bit, and a very small amount, okay? And so that little bit of enolate is still a nucleophile, but it's in a large sea of what? What's there in excess? The ester itself. That's exactly right. So Matthew knows that we generate just a little bit of enolate. So we have 99% of our solution is this, and about 1% of our solution is the enolate. Are esters good nucleophiles or good electrophiles? Think about everything we've done to an ester before. Has they functioned as an electrophile or as a nucleophile? Think about some of the chemistry we've done with esters. What have we done to esters? We have made them carboxylic acids. What else have we done to them? What happens if we throw in lithium aluminum hydride? We reduce them. And the hydride's acting as what, a nucleophile or an electrophile? A nucleophile. So esters are behaving like what? Electrophiles. So I have just generated a little bit of a nucleophile in the presence of a large excess of electrophile. So what do you think happens? They react with one another, right? So let's work through the mechanism. So we've got at least 2 moles of ethyl acetate. We throw in a mole of sodium methoxide, and then we work it up as typical by dumping into mild acid. Okay? So we've just generated a little bit of the enolate. What happens? How does it react with the large excess of electrophile? Sam, help me push the arrows.
Let's see, the pi bond is going to the carbon on the left. It's going to a deck. The carbon.
It is. I need to push one more arrow, though, to do that.
The negative charge 1 there is going to come down and attack that.
And this is going to then attack, what did you say?
The carbon.
Okay, and now what happens?
The pi bond on that goes up to the oxygen.
All right, and so we end up getting a tetrahedral intermediate, that's right. So this part comes from what? My nucleophile or my electrophile? The nucleophile. It came from the enolate, right? This part comes from the ester that's functioning as the electrophile, okay? So now, Rose, what happens? Yep. And so we get now what happens? Looks like we're done, but we're really not done yet. We haven't dumped it into acid yet. So what happens now? I've got a base. Do I have a strong acid, relatively speaking? That's right. These hydrogens. And why these hydrogens and not these hydrogens? And why are they more acidic? Very good. So the base comes in and abstracts one of these hydrogens. And so I get ethanol as a byproduct, plus I get this anion, oops, that's OET, excuse me, which stays perfectly happy until the reaction's over and we dump it onto acid and it just gets reprotonated. This is how you make beta-keto esters, and it's called the Claisen reaction. It is nothing more than the reaction between an enolate of an ester and itself. The mechanism's pretty straightforward. The enolate reacts with the carbonyl, you get the tetrahedral intermediate, and that collapses and you get the product ultimately after workup. Uh-huh.
You don't even get much of that product anyway.
This product? Yeah. It goes well.
Oh, I thought you meant like 1% of that.
You do get 1% of this. But as it's consumed and reacted, what happens? Le Chatelier's principle means that more of this will form, right? So, is this reaction catalytic in sodium methoxide or stoichiometric? It is stoichiometric because once we generate, it looks like I'm using sodium methoxide, but I get ethoxide, right? But that immediately reacts with the acidic hydrogen and gets consumed. So you have to have a full equivalent of sodium ethoxide. Notice what base we are using. The base that we use, sodium ethoxide, the OET matches the ester part. So if we were using methyl acetate, what would our base be? Sodium methoxide. Can anybody tell me why we want to use the base that is derived from the alcohol that's used to make the ester? What would happen if we did this reaction in sodium methoxide instead of sodium ethoxide? Would the reaction still occur? The short answer is yes. It will occur. We will get, but we won't get this ester as the product. It'll undergo what's known as a transesterification. Because methoxide can attack the carbonyl, right, and kick out ethoxide as the leaving group, and then you end up with the methyl ester. And that's an equilibrium, and it gets messy. So to avoid that, we always use the base that is derived from the alcohol that's used to make the ester. Why is that? Because you can't tell when that reaction happens. Sodium ethoxide can do the exact same chemistry I just described for the sodium methoxide, but it gives you the same product as you started with. So it's kind of a slick way to avoid transesterification. Okay? So, what do we need to know? We need to know that the Claisen reaction does what? Why is it important? What is it forming? New carbon-carbon bond. And what is the product? A beta-keto ester. And those beta-keto esters are actually important molecules for example, in the medicinal field. And they're easy to form. Okay? You can also do what's known as a crossed Claisen reaction. Why is this called a crossed Claisen reaction? Look at that and tell me why it's a crossed Claisen reaction. I've got two different esters. What can you tell me about these two different esters? Okay, they're both ethyl esters. Good. What else can we say about them? What does one have that the other doesn't? Pardon? A ring. And what else? One of them has alpha hydrogens. The other one does not. Right? That is important. So which one of these forms the enolate? It's the one that only contains the alpha hydrogens. Okay? And so you end up with this beta-keto ester as the product. Very, very important to note. Okay? What other product might you expect to get from this reaction? So Gabby comes in and runs this reaction, and she finds that product, she gets a 95% yield, and then there's a 5% of some other keto ester. What do you think that other 5% is? That's exactly right. We're not going to generate completely the enolate from ethyl acetate, right? So once she's generated a little bit of the enolate, it can either react with itself or it can react with the ethyl benzoate. Can anybody tell me why it would prefer to react with ethyl benzoate over ethyl acetate? It turns out that the carbon of that carbonyl, because of the resonance with the benzene ring, is more electrophilic. And so it reacts preferentially, but not solely. Okay? So, let's do a little experiment here. Let's say Katie wants to do a crossed aldol, or not aldol, excuse me, cross Claisen between these two esters. What's the product? Or what's the problem? Let's start off with the problem, not the product. That's right. I have got two esters that both have alpha hydrogen, so I'm going to generate how many enolates? Two different enolates, right? I'll generate the enolate here, and I'll generate an enolate here. That's three? Let's think about it. So we'll call this A, and we'll call this B. So if the enolate is A, it can react with itself, which would give me AA, or it can react with what? AB. Now, B as an enolate can react either with itself, right, or A. Those are four different products. We can generate four different products. So if we're lucky, at best, we would generate the product that we want in what kind of yield? 25%. If you go to work for an employer, that'll get you fired. Okay? So if we wanted to generate, well, we wouldn't want to generate that one. Let's say we wanted to generate BA. The best we could hope for is 25% yield. Okay? I want you right now to draw those products for me, and then we'll look at them. Work with your buddies if you want. What would the structure of those four products look like? Goodness. So what does AA look like? Like the one we did before, exactly, right? So it's going to look like this. Okay, where A was the enolate and A was the electrophile. What's AB going to look like? Well, that means A is the enolate, right? And what is the nucleophile? Or the electrophile, excuse me? This part, right? So this is AA. This is AB. Will everybody agree those are clearly different products? All right. Now what about BA? Well, B is the nucleophile or the electrophile? It's the nucleophile, right? So it's going to be the ester part of my product. Right? And what's, oh, and BB. Did them out of order. And so we have four possible products in terms of carbon-carbon bonds. Is that all the products we're going to get, though, if we look at those? Are there any stereocenters here? Are there any stereocenters here? No. Are there any stereocenters here? So this one, we're also going to get a pair of enantiomers, right? We're going to get R and S. And how about this one? We will also get a pair of enantiomers, R and S. Will those enantiomers be R favored, S favored, or racemic? They will be racemic. That's right. So you'll get a 50-50 mix of both R and S. So actually we end up with 6 total products if we take stereochemistry into. Pardon? Which isn't good, right? But in the crossed Claisen reaction, because only one ester has the alpha hydrogens, the other one can't generate an enolate. This one can only function as an electrophile because it has no alpha hydrogens. So we know that if we just generate a tiny amount of this ester, it can either react with itself or it can react with the ethyl benzoate. How could we avoid that 5% byproduct that Gabby found? What could we do to get this product as 100%? In other words, what's the problem with as it's written? Sodium ethoxide only generates how much enolate? What do you think, Russell? How much enolate does it generate for me? A lot or a little? Very little, right? So since I generate very little, it can either react then with itself or it can react with the ethyl benzoate. And we're depending on the ethyl benzoate being more electrophilic to give us mostly that product. What stronger? Okay, so generate the enolate completely, right? Is a solution to this problem. Sarah, how can I completely take ethyl acetate and generate the enolate? What base would I use? What do you think? LDA. That's right, that'll work. So she would come in, she would take a solution of LDA, she would cool it down to minus 78 degrees. She would add ethyl acetate dropwise to that to completely generate the enolate. And once we've completely generated the enolate, it can't react with itself, right? Because it's all enolate. And then she would, at low temperature, start to add the ethyl benzoate in slowly to keep the temperature cold, and the reaction would go quite nicely. That's called a directed Claisen, where we're completely generating the enolate from one species. And that's how we can actually do this chemistry if we want. So let's suppose we want BA completely. We don't want this 25% stuff. Let's not pay attention to the RNS stuff, okay? Let's say Matthew wants BA completely. What he's going to have to do is completely generate the enolate from B first using LDA. Once he's completely generated that enolate, it can't react with itself. So what does he do? He then adds the ethyl acetate, and that means that this is going to function solely as the nucleophile. This will act solely as the electrophile, and he will get this product pretty much so exclusively through this directed Claisen reaction. Okay? This way, well, if you were to use these conditions, I should say, you're going to get a statistical mixture. Okay? Because we're not generating enough of the enolate, okay, at once. Does that make sense to everybody? It makes sense when I'm talking about it, but when you go and do your homework, let me know if it makes sense, okay? So that's the important thing. So it makes carbon-carbon bonds and beta-keto esters, okay? Keep that in mind. So let's look at some examples. These are things from the old ACS standardized exams. What would be the major product of this reaction? The first thing that you need to do is look at the starting material and the reaction conditions, and you automatically know it's what kind of reaction? You know it's a Claisen reaction because you have esters and a base, right? So we know it's going to be a Claisen reaction. And we know that a Claisen reaction is going to generate a beta-keto ester. Which one of those products up there can we eliminate based on that information alone? You can eliminate D, A beta ketoester it ain't, right? And what else can we eliminate? A, because we have what? We actually have a diester, right? So we have an ester and an ester. And we can eliminate C. So based on that information, we know that the answer has to be? That's right. Most of you, when you get on these standardized tests, forget how to do this kind of stuff. A lot of time it is just a process of elimination. And it's not cheating. It's knowing, hey, I know that that's going to be a Claisen reaction. I got to get a beta ketoester. What ain't a beta ketoester? Right? And by the way, this is how doctors think. So for those of you who want to go to medical school, you come in, you got a fever and your chills. There's tons of things that have that, right? And they go through process of elimination and then decide what to prescribe you, which is an experiment, by the way, right? And then they have you come back and they check up on you to see how their experiment went, okay? But it's this type of thinking that's important, okay? So because we knew that we were going to get a beta ketoester, we could figure that problem out without too much issue. You didn't need to remember the mechanism, right? Now, you know, I'm a big stickler. I like for you to know the mechanism. But you could have got this question correct on the ACS final exam by simply knowing your reaction types, okay? How about this one? What kind of reaction is this? It is a cross-Claisen. And again, we know that we're going to get a, what's the product? Beta ketoester. So what can we eliminate based on that alone? We can eliminate C and D. You know, you just went from a 1 in 4 chance to a 50-50. You know, if you have 50-50 odds at something, you always put your money on the table. The odds aren't against you, right? They're not necessarily for you either, but they're not against you. 1 in 4 chance, the odds are actually against you. So it can either be A or it can be B. Which is the correct answer? It is B. It is B. So we know that this molecule is going to function as the enolate, right? And that gets generated at the alpha carbon. So all this is going to be this part here, right? So this methyl group is attached to the alpha carbon, and then this part comes from our ester without alpha hydrogens. Does that make sense? But again, immediately knowing that we have to get a beta ketoester, we can eliminate C and D. We could give ourselves a 50-50 fighting chance, okay? Very, very important that you take these good test-taking skills with you into the final exam, okay? Because you're going to have 70 questions on the final exam. We will have covered everything that's on that final exam. You will know everything you need to know going into that final exam. But what gets most students is they forget good test-taking skills, okay? I've seen students that I know the answers that don't get them because they panic, they don't take their time, they don't eliminate. Okay, very important that you eliminate absolutely incorrect answers right from the bat on every question. Okay, now there'll be some questions where you go, well, I really can't eliminate anything, and you're going to have to think a little harder. But I'd say 25-30% of the questions, you're going to be able to eliminate at least one as total nonsense, okay, if not two. So you'll be able to do it. How about this reaction? Which of those reactions listed there, 1, 2, or 3, can form that product? I'm asking you all to think about it. 2 and 3 can prepare it. 2 and 3 can prepare. You're just reversing the roles. Right? Now, you'll be looking at this going, well, this is a ketone, it's not an ester, and that's a carbonate, that's not an ester, but nonetheless, it'll work. You generate an enolate here, we can generate those from ketones. It will attack here and kick one of these out as a leaving group, and you end up with the product. Okay? Haven't told you that one specifically, but mechanistically, you could have figured that one out. Okay? The Dieckmann condensation is another name you need to know, but you don't need to know anything new, because it's nothing more than an internal Claisen reaction, and it forms rings. That's the important thing about it. Notice the molecule contains 2 esters, and using a base, we will do a Claisen reaction to get a beta ketoester out of it. But as we generate that beta ketoester, we also form a 6-membered ring, and that's why it deserves its own name. Dieckmann recognized that you can form 6-membered rings using this reaction. Can anybody tell me why that's important? Rings are useful? What about them? They're very prevalent in biological systems. Look at a steroid structure sometime. 6-membered ring after 6-membered ring connected together. Okay? You just have the 2 esters that are connected together. You can form a 6-membered ring through the Dieckmann condensation. Now, you can also form 5-membered rings if you have one less carbon in between the 2 esters. Okay? But it's well known for its ability to form 6-membered rings. Okay? What else do we know that readily forms 6-membered rings that we learned about? The Diels-Alder. But the Diels-Alder will produce a 6-membered ring with an alkene within the ring, right? Is there an alkene within this ring? No. This allows me to build a 6-membered ring and have an ester and a ketone that I can do other things with. So maybe Jamie wants to make this 6-membered ring because it has a ketone, and she wants to convert it to an alcohol. So she does this reaction first, and then what does she do to reduce this to an alcohol? What reagent would she use? Remember, we got a ketone and an ester, so we have to be careful. What reagent will reduce a ketone over an ester? Sodium borohydride. That's exactly right. Or maybe Jamie wants to leave the ketone alone and reduce the ester. How can we do that? We got to have to protect something. And where are we going to protect? Ketone. And how are we going to protect the ketone? Make an acetal or a ketal, right? So she would take it, make the acetal or ketal, then reduce the ester with what reagent? Lithium aluminum hydride. Sodium borohydride is not strong enough typically to reduce an ester, right? So she'd use lithium aluminum hydride, reduce the ester to the alcohol, and then how does she get the ketone back? Add a little acid and water, right? And it will remove the ketal and replace it with the ketone. Remember, that's an equilibrium process. Okay? From this point on, actually from chapter 18 on, you're learning no new mechanisms. We've already learned everything that you really need to know. Right now, we're just putting them in different orders to do new things. The Claisen reaction isn't new in terms of mechanism. The Dieckmann isn't new in terms of mechanism. Okay? They're all just based on enolates. So which carbon that's shown up there in the starting diester served as the nucleophile, and which one serves as the electrophile? I've got them numbered for a reason. Which carbon, which numbered carbon acts as the nucleophile? One functions as the nucleophile? 7 functions as the nucleophile? 6. Eventually we'll get there if we go through them all, right? 6. And how do I know 6 is the nucleophile? Because this is the part that had to be the enolate, because that's where the ester is. Remember, the ester becomes the enolate, and that functional group is retained. This ester, right, is no longer there. But carbon number 1 is. The carbonyl of that ester is there. So we generate the enolate here. That enolate then attacks this carbon of the carbonyl. We get the tetrahedral intermediate, which then collapses to the ketone. Okay? And in the process, we form a 6-membered ring. Okay? The reaction for this is about 80-90% yield. I actually got this particular reaction out of a journal article. So that's isolated yield. Very easy to do. You take the diester, you throw in, what base would you use? I wrote base up there, but what base would we use? Chance, what do you think? What base would we use? Would we have to use LDA? You could. That'd be overkill. Pyridine's not strong enough. What's the ester? What's the alcohol of the ester here? NaOCH3, sodium methoxide. That's right. We've got the methyl esters. So you would just use sodium methoxide in methanol as the solvent, and you would get this product. Now, could we get the product where 2 functions as the nucleophile and 7 functions as the electrophile? Yeah, but could we tell any difference? And why not? It's symmetrical. That's right. That's why we can get away with just using a base like sodium methoxide, because we don't have to worry about which carbon serves as the nucleophile, because it's symmetrical. We're going to get the same product either way. Okay? So what is the starting material that will yield this compound upon treatment with sodium ethoxide in ethanol? Yeah, they don't always do that, though. Anything up there that we can eliminate? What were you counting, Gabby? What carbons? Okay, so if you look at this, what we know is that we're forming a 5-membered ring, right? So between the carbonyls, we have 1, 2, 3, 4 carbons. Find the carbonyls and count the carbons in between them, and that'll help you. D is the right answer. So we generate the enolate, right? And then that attacks here. And so in doing so, we're going to form 1, 2, 3. There are going to be 4 carbons in between those carbonyls. So in a question like that, count the carbons in between the carbonyls, and that helps you identify which one can or cannot be the correct answer. How about this? Wait a second. We haven't talked about beta diketones, have we?
Well, we can figure it out.
But you can figure it out. You all are smart. So let's figure it out. What's that? What kind of ring is being formed up there with the 2 ketones? It's a pentane ring, yeah, 5-membered ring. C is the correct answer. So we can generate the enolate from this ketone, right? That then will attack the carbon of the carbonyl of this ester and give me my product. So it goes through the tetrahedral intermediate, that leaves. If I use this, I would end up, the enolate would attack the ketone, and I'd end up with a tertiary alcohol, not the product. Here, I would end up with what? If this enolate attacked this aldehyde, what would I end up with? Not a ketone, because I have no leaving group. So what would I end up with? What kind of alcohol? Secondary alcohol. That's right. And then for D, where could I generate the enolate? Here, right? So if that attacked here, would I get the right size ring? No, and in fact, it would be a very strained ring, because it would be 1, 2, 3, 4-membered. And actually, that reaction wouldn't occur at all because of the ring strain. Okay. You're going to see things like this that aren't explicitly covered in a chapter per se, but the mechanisms are there. Okay? You can figure it out at this point. Generate the nucleophile, the enolate, look for the electrophile, see how the chemistry goes. Okay? Now, we can use malonic esters. We talked about this a little bit last time, so I'm not going to spend too much time on it, but malonates, like diethyl malonate that you see there, those hydrogens are particularly acidic because between two esters, between two carbonyls. What's the rough pKa range? Let's just do some simple math. If the alpha hydrogens to an ester are around 25, what do you suppose those alpha hydrogens are going to be between two esters? Yeah, 12, 13, somewhere in that ballpark, right? So, pretty acidic. We can generate new carbon-carbon bonds with them using enolates, and then we can decarboxylate them and get carboxylic acid. So it's another way of making carboxylic acids, okay? And so we know that diethyl malonate, for example, or dimethyl malonate, it doesn't matter. If you heat it up in acid, it hydrolyzes the ester to the carboxylic acid. And once you have these diacids, they like to lose carbon dioxide through this 6-membered transition state that we've talked about before. Okay? And so you can form new carboxylic acids with them fairly easily. So let's look at an example. This is an example similar to one that we've actually done in the lab. So you start off with diethyl malonate, which is dirt cheap, by the way. You pay more to ship it to you than it costs, which always kind of frustrates you, but it's cheap. You treat that with sodium ethoxide, you get the enolate. I'm showing the negative charge on the carbon, but we know that the resonance forms exist. I treat this with an alkyl halide. What kind of reaction is that going to be? SN2, right? So we form a new carbon-carbon bond right here. And then we dump this into H2SO4 water and heat it up. We will hydrolyze the esters to the carboxylic acids, and then a decarboxylation occurs, and we end up with this carboxylic acid. Okay? So we will pick up with this on Wednesday. What else is due Wednesday? Your extra credit. That's right. No excuses for it being late. So if you think you're going to be gone or you're feeling a little peaked, it's a good idea to go ahead and turn it in. Okay? Have a good one.